Thank you so much, everyone, for coming. I'm delighted to be here with Professor Matt Meselson of Harvard University. I'm David Zieler, director of the Caltech Heritage Project, which is Caltech's institutional history program, and I'm here to engage Matt about some deep Caltech history from the 1950s. So Matt, to start, you grew up near Caltech. What did you know about Caltech, even as a high school student?
Well, kids in even junior high school and high school were very aware of the existence of Caltech. And I don't know the first time I came here, but it was sometime when I was in high school. And I worked for Eddie Lewis, the geneticist, fruit fly guy. My job was virginizing female flies. So if a Drosophila female has mated, if you subject her to low temperature, all the sperm in her body are killed. And so you can use her for an experiment more than once. That is, you can make more crosses than one with the same female Drosophila. I don't know why Lewis bothered doing that. There was no shortage of flies. I guess that he had a few genotypes of which he didn't have too many flies. And this, or maybe he was just being very stingy. But that was my job. It made all my friends laugh that I was virginizing females.
Matt, did you consider Caltech as an undergraduate?
I was an undergraduate here. I was a freshman. But it was after I had finished. It's a long story. Okay, I'll give you the whole miserable story. So during the war, a lot of us felt that we should be doing something other than just goofing off. And so I took summer school courses and I worked in a defense factory. And because of the courses, I had all the academic credits I needed to be given a diploma, a high school diploma. I was going to John Marshall High School over near Franklin Avenue in Los Angeles, almost over to Hollywood. So I went to the registrar at John Marshall High School and said, I'd like my diploma. And she said, you have to have, according to California law, 3 full years of physical education. And you can't get your diploma without that. But I had all the academic credits. I asked her, what am I supposed to do? She asked me, were there any courses I liked in high school? I said, yeah. She said, well, you could take them over again, like sort of ice cream. Or this really shocked me because up to that point, I thought that everything had been arranged very wisely by the grown-ups. And to think that you'd have to spend an extra year running around the track boggled my mind. And I asked around and I learned there was a place you could go without a high school diploma called the University of Chicago. So I went there and got a bachelor's degree. They didn't give bachelor's degrees in special subjects, only in the classics. You had to read not up-to-date science, but for optics we read Fresnel. And for biology, we read Harvey on the circulation of the blood. And for physics, we read Galileo, 2 new sciences. We read all these old papers. And I'm glad I did, because I would never have read this stuff otherwise. But at the end of that, I went to Europe for a year. And then I had to start over again in science. I came here to Caltech. And I liked it very much. I especially liked Pauling's chemistry course. But there was one thing that irritated me. In the physics, you know, you can derive the conservation of angular momentum from the conservation of linear momentum. But at that time at Caltech, the conservation of angular momentum was taught as a separate thing to memorize. And I thought that was just awful. And it burned me up so much, I decided to leave Caltech and go back to the University of Chicago, which I did. So I was only a freshman here. But then I did some other things. I started out in physics at Berkeley, but the class sizes were huge, like couple hundred people in each class. I didn't like that. So I was going to go back to the University of Chicago in physics. But while I had been a freshman at Caltech, I got to know Linus Pauling's daughter, Linda Pauling. And Linda invited me to a party at her dad's house. He had a big swimming pool. It was a daytime party in the swimming pool. Everybody was swimming around. So I'm in the water, and Linus Pauling, the world's greatest chemist, comes out of his study. He knew me because I'd done a little research project as a freshman for him that year I had been here. He looked down at me in the water and he asked, well, what are you going to do next year? And I said, I'm going back to the University of Chicago. And he said, oh, that's a lot of baloney. Come be my graduate student. So because of this accident in the swimming pool, I came back to Caltech. And then I stayed a long time. I was a graduate student. I was a postdoctoral fellow. I was an assistant professor. I taught physical chemistry for 2 years. But I couldn't get much laboratory work done. For me, at least, I spent too much time preparing coursework. So I asked George Beadle, who was the chairman of the biology division at that time, if Caltech would consider demoting me back down from assistant professor to postdoc, because then I'd have all the time in the world in the laboratory. And I think it's greatly to the credit of Caltech. I mean, every university promotes people, but Caltech had the wisdom, of course, at a higher salary, but nevertheless to demote me, to take me off the usual academic track. I didn't have to be an assistant professor anymore. And then I went to, after 2 years of that, I went to Harvard. I wanted to see what the East was like, and I stayed there until now. I mean, here I am. I haven't been, well, I guess I was here once.
Matt, when Linus convinced you to come to work with him,
he didn't have to convince me. As soon as he said that, I said yes. And I remember, which could be inaccurate, but here I'm in the water, and I said yes. And I still have this feeling of becoming less buoyant, because when you expel some air from your lungs, your body volume decreases slightly, and you sink because your weight doesn't decrease. And I still have that feeling when Pauling said, come be my graduate student, and I said, yes, boom, like that.
So you knew you would work in the chemistry program. Did you already have interests in biology?
I always wanted to know. I had a big periodic chart at home. How is it you can put together some of those elements and have something that's alive? And I thought initially it had to do with electricity. And the reason for that is that in Michelangelo's painting of God and Adam, and here's God up here and Adam here, and their fingers nearly touch, there's a little space in between. And so since I knew, as you know, that if you have a charge on an object, that if you have a point where the radius is very small, that's the point where the field will be strongest. And so I imagined, although there's really no such thing in Michelangelo's, I don't even know if he knew about electricity, maybe he did. There was really no spark there, but to me there had to be a spark. And for this stupid reason, I mean, I was a really little kid when I got this idea from the painting of Michelangelo, that it had to do with electricity. So I became interested in electrochemistry, which meant, in particular, I was trying to prepare various metals by electrolysis in the chloride salts. You can, for example, the one I tried hardest and never could get was europium. It's a rare earth metal. So I had europium chloride molten and 2 electrodes, a bunch of batteries, and I was hoping to be able to make metallic europium. It never worked. So I did all that stuff when I was a very little kid. I had a big laboratory. I had a furnace and analytical balance and a spectrometer and all kinds of stuff. And I purified rare earth oxides and sold them to a company in New York. And then they resold them. I didn't realize at the time, but I was selling samarium oxide for something less than $1, and they resold it for like $10, because I went out to New York and actually met the guys at that company. It was called the A.N. McKay Company, and saw that which was perfectly appropriate. That they should charge whatever they want if people would pay for it. So I was very interested in rare earths, a very interesting group of elements. You know, most elements, as you go from one to the next one higher in the periodic chart, you're adding electrons to an outer shell. And since it's the outer shell that determines most of the chemical properties when you go from element atomic number n to n+1, it's a really different, chemically different element. I mean, you're going horizontally on the periodic chart. So, but in the rare earth elements, as you go from one to the next higher one in molecular and atomic weight, the electrons that are being added to an inner shell, the 4f shell. And so the outer shell is the same in all the rare earth elements. And so they're all chemically very similar, even though they're in a whole horizontal row in the periodic chart. So, but they're another property, I don't want to bore you too much with this, but since when you shine light on them and it's absorbed, the absorption is happening in the 4f shell. There's a transition there. So the outer electrons are not influenced when you shine light on them. So that means that the atom, when it's in solution, is not being hit by water molecules in any place that would change very much the energy of the transitions of absorbing light. So what I'm saying you should see already, that means that whereas the absorption, say, of manganese, which is purple, manganese salts in water, if you look with a spectroscope, it's a rather big fat band. But in the rare earths, the absorption bands are very, very narrow, and that makes their colors, and this is what I'm getting to, really gorgeous. There's something that painters call hue, which is the purity of a color. Is it really just a single wavelength? I mean, most colors aren't, like this guy's shirt, sort of blue, but if you had a spectroscope now and looked at his shirt, it'd be a fairly fat band in the blue. But if it was a rare earth shirt, I don't think you can get a rare earth shirt, but it would be a sharp band in the blue. But that's not what you came to hear about.
Matt, what was Linus working on when you became his graduate
Well,
student?
he was working on the alpha helix, and he got the alpha helix in bed.
I wonder if you can explain what the alpha helix is.
So Linus became interested in the structure of proteins. He wanted to apply, especially structural chemistry, to biology. And there's a particular protein called feather rachis hormone protein. It's one of the proteins in bird feathers. And you could make lots of it, and it was nice. You could crystallize it. And he was trying to get the structure of that. But by the time that I came back to Caltech, I think he already had the alpha helix. It's a structure he proposed for all protein molecules, in which, see, all protein molecules have a peptide group in them. And so there's a kind of, if you imagine a protein with no special side groups, like polyalanine, he designed a molecule. Okay, I'll tell you the whole story. He was in London, and he got sick. I don't know what it was. I think it was nephritis, but it was something. So he's in bed, and he's still thinking about how to make the structure of, how to determine the structure of a protein. And he was wrapping paper around dowels of wood in bed in the hospital. And this way of getting a helix on a piece of paper, you wind the paper around like on a broomstick. And he tried various ways of connecting up the atoms of the peptides, and that's how he first found an alpha helix, or at least the idea that led to the alpha helix. So, but when I got here, I'm not sure what it was. I can tell you some funny stories, but he was still very interested in hemoglobin. He gave me a project as a freshman. If you heard my talk yesterday, I'm just repeating myself, but he was interested in the hemoglobin of all kinds of different creatures, including a big worm that lives in the sand at the Caltech Marine. Does Caltech still have the Marine Station?
Yeah.
at Corona del Mar. You should go there if you haven't. Great place. And can you still sleep there?
Oh, I don't know.
Well, students used to be able to, they had bedrooms, a few bedrooms, and you could just sleep on the floor in a sleeping bag. Anyway, this was an interesting worm. Its hemoglobin was different in some ways from other hemoglobins. Linus wanted to know what the size of the opening was through the protein leading to the heme group, which is a group that does the gaseous exchange. And so how do you determine if you can't do X-ray crystallography on the hemoglobin of this worm, how do you determine how big the hole is from the outside to get into the heme, which is inside the protein? Well, his idea was alkyl isocyanides bind to the iron in heme and change its color. But if the isocyanide is too big to go through the hole to get to the heme group, you're not going to get any spectral change. So my job was to synthesize a whole bunch of alkyl isocyanides, methyl, ethyl, isopropyl, butyl. I think I got up to butyl. And I was supposed to then look to see which ones would make a spectral change, meaning they got to the iron, and which ones were too big to cause a spectral change. I never did any of the spectroscopy. And at the end of the year, I went to Linus thinking, now I'm going to catch hell. But he then said, Matt, you've learned what I wanted you to learn from this, which is that it takes a lot longer to do research than you think at the beginning. And he was always just so nice about everything. Yesterday did I tell about
what?
Pauling and Plato?
We talked about that.
I told that story? Okay. What other Pauling stories do I know?
Did he have interactions with the biologists? Did he talk with Delbrück?
Yes, he was a good friend of George Beadle, who was a geneticist, and Max Delbrück, who was in biology, was a physicist. And so Linus, who had studied quantum mechanics with Sommerfeld in Munich, wrote a book on quantum mechanics with one of his students, E.B. Wilson. So Max and Linus knew each other. I don't know that they were close friends, but when Linus wanted to write a paper suggesting how genes duplicate, he went downstairs because Max's office was in the basement of church and asked Max if he would sign it. So it would be two people, which Max did. So there's a famous paper by Pauling and Delbrück. I don't know about how personally they were with each other. Pauling had some good friends on the faculty, but I don't know enough about that.
Did Pauling react to Watson and Crick's paper in 1953?
Well, that's very interesting. So Pauling had been working on the structure of DNA at the same time that Watson and Crick were. And he came up with a three-stranded structure, which is wrong. Now, why was it a three-strand structure? One day, a man named Robley Williams from Berkeley came to Caltech and gave a lecture. He had, with the electron microscope, looked at DNA molecules and measured their diameter. Pauling had gone to that seminar, and you can read this. All of Pauling's notebooks are on the web. You can just go find them on the web. And the notebook for a certain day in November of 1952 has the whole story. And in it, he says, Robley Williams says that the diameter is 17 angstroms. Now, Pauling knew that probably in a fiber, it was known that DNA molecules are long, that the molecules would pack in the most compact way, which for cylinders is cubic closest packing. Now, if you slice something which is a bunch of cylinders in cubic closest packing and draw lines between the centers of the closest cylinders, you'll see that you have a shape which has 60 degrees here, 120 degrees here, 60 and 120. So you know the angles of the unit cell. And you know the dimensions this way because that's where two cylinders are touching, that's twice 17 angstroms. So you know the area of the unit cell. And person in England had measured the repeat distance along the DNA molecule. And so knowing that length and the area of the base, multiply them, you've got the volume of the unit cell, right? Now, Alex Rich, who was a postdoc here, had measured the density of a DNA fiber. To do that, he took a very simple graduated cylinder, and you pour carbon tetrachloride in it, and then very gently pour some ether or some light hydrocarbon liquid on top, and let it diffuse a little bit, and you've got a density gradient, because carbon tet is dense, ether is quite light. And then you drop your little piece of whatever it is you want to measure the density, in this case DNA, and you see where it comes to rest. And it'll come to rest at a place where its density matches the density of the liquid just next to it, buoyant density. So Alex had done this, and he measured the density 1.72, I think it was. So density is mass divided by volume, right? So Pauling had the volume, as I just told you. So this made it possible to know the mass of DNA inside of the unit cell. Are you following me? Good, okay. So now he knew the mass of a single nucleotide because we knew the chemical structure of it. And so he then asked, how many of those nucleotides do you have to stuff in to this unit cell to get a density that Alex Rich had measured of 1.72? And the answer was 3. Now let me digress to Jim Watson's book called, what's it called? Jim's famous book. Everybody's read that book. Anyway, in this book, Jim relates that he and Francis had, with atomic models, and of course the whole idea of making models of atoms in order to exploit molecular structure like a game came from here, because Pauling, Caltech had a terrific shop, maybe you still do, and the whole idea of beginning to make accurately sized atomic models so that you could put them together and design molecules, in effect, all started here. So Watson and Crick had used models, ones not made at Caltech, but made in the shop at Cambridge University, and they built a structure which they were very proud of. It was a 3-stranded structure. This is all in Jim's book. And they were so proud of it, they called Maurice Wilkins, who's down at King's College in London, also working on DNA. And Rosalind Franklin was also there, working on DNA. Rosalind decided she wanted to come up with Maurice in the car to see this wonderful model, Watson and Crick. So they come up there and they see the model, and according to Jim's book, Rosalind says, "You forgot the water." It was a 3-stranded molecule, just like Linus. And that's what Linus had forgot too. There was more mass in the unit cell than just nucleotides. There was a little water. And so the arithmetic that I just went through with you, you do need the mass that you calculate from the density and the unit cell volume, but the mass isn't nothing but nucleotides. It's nucleotides plus a little water. And that's what caused Watson and Crick to build or to put it simply, if Rosalind Franklin had been at Caltech and looked at Linus's model, she might have said, "You forgot the water." And he might have got the right structure. And there's another reason he might have got the right structure. Linus had so many ideas all the time, but then he'd forget them. So he gave a lecture in England in 1948. How might a gene replicate? It's incredible intuition. The title of the lecture was "The Role of Chemical Complementarity," which means the various ways in which molecules can fit together. Van der Waals forces, hydrogen bonds, electrostatic bonds, all that kind of stuff. About how those things could apply in biology, like antigen-antibody interactions have to do with the detailed complementarity of the the structure of the antigen and the antibody, they fit together, so on. So he asks in this, it's a published paper, how might the gene replicate? And then he says, if the gene is made of two parts, which are complementary to each other, and the two parts separate, and on each a surface of each, a new chain is made, that's how the gene might replicate. It's exactly right. That's how DNA replicates, except this is 1948. We didn't find about semi-conservative replication of DNA till 1958, 10 years later. But Pauling, if he had remembered this, he wouldn't have proposed a three-strand structure, because that's not two things fitting together like this. The three-strand structure was totally, and this brilliant idea he had about how genes replicate, he forgot it. So therefore he proposed a molecule that was wrong, a three-stranded molecule. But the story is very complicated and very rich, and it could have turned out different. And it's a wonderful story.
Matt, when did you start interacting with Delbrück?
With who?
Delbrück.
Well, in those days, I hope it's not true now, the mathematics students at Caltech, at least I thought they did, looked down on the physicists. And the physicists looked down on the chemists. And the chemists looked down on the biologists. I can remember that as a chemist, I was in chemistry. To me, the biologists mainly were guys who did slimy stuff. I sort of looked down on them in that sense. But of course, Max Delbrück, he was a quantum mechanical physicist. And I wanted to apply chemistry to what makes things alive. And so I needed to talk to some kind of a biologist. But the only guy in the biology division, do you still call it a division? I remember you weren't allowed to call it a department.
Same.
It's a division. Is that still the case?
Yeah.
I don't know why there's so much feeling behind. What's wrong with calling it a department?
It's a Caltech idiosyncrasy.
Linus had a reason for this, and it had something to do with his feeling about the breadth of his science. Anyway, I don't want to get off onto that. So, what was I talking about?
How you got to know Max.
How I got to know Max. So I needed to talk to a biologist. But the only biologist I thought I would be able to talk the same language was Max. Max Delbrück had a fearsome reputation. For example, if in a seminar he thought that somebody was saying something stupid, he'd say, "That's stupid." You know, it was horrible, not very polite. But he was not a power-hungry guy. He had a little tiny office. So I went to see Max. And this was just after Watson and Crick had published their two papers in Nature. So it would have been like May or April 1953. And I'd never heard of Watson and Crick or the double helix or any of that stuff, even though I should have heard of it, but I hadn't. So the first thing he says is, "What do you think of these two papers by Watson and Crick?" I said, "I'd never heard of them." Well, he had a big stack of each of these because Jim had known Max earlier from Cold Spring Harbor. They were great pals. And so Jim sent Max a whole bunch of reprints, not just one. So that's why I had a little stack of each one. He picked these up and he threw them at me like that. And they sort of fluttered down onto the floor. And then he shouted, "Get out and don't come back until you've read them." And what I heard was, "Come back." So I went back. It's a long story, but did I tell this story the other day?
Yeah. What were some of Max's misgivings about Watson and Crick's?
Well, these are long molecules. They're actually much longer than we even thought in those days, but even then we knew that they were pretty long, from flow birefringence measurements and things like that. And so the way that Francis and Jim had proposed the molecule replicates was a complete unwinding of the whole molecule. So there's only two ways to do that. One is you hold the two arms fixed and rotate the bottom cylinder, or you hold the bottom unreplicated piece fixed and you rotate these arms like that. Well, either way, and especially this way, does a lot of work against the viscous drag of the liquid in which it's present. And you can calculate, even if it was water, there's an enormous amount of work that you have to do to unravel a really big DNA molecule. So Max didn't like that. So he proposed that every 8 bases, something would break a chain and allow swiveling around the remaining single bond and then reseal it. In other words, he had imagined topoisomerases before anybody knew that there were such things, except he didn't have any idea how this would happen. But basically, that's, if there was a topoisomerase that worked every 8 bases, then you could unwind the two by breaking the chains and resealing them without having all this viscous drag. So he published that together with Gunther Stent. And when he proposed that, the second time I went to see him, the time that he said that when I went back, I had a different idea of how to find out. Did I tell this story also?
No, go ahead. Please.
Well, I've been taking Linus Pauling's quantum mechanics course. I don't think it was called quantum. I think it was called The Nature of the Chemical Bond, but mainly it was some quantum, very kindergarten quantum mechanics. And one of the problems was to calculate the relative energy of the hydrogen bond and the deuterium bond. It turns out you can do that. It's a one-line proof from the quantum mechanical zero-point energy. So once you see it, it's really simple. And I was really impressed with this, that you could calculate the ratio of the bond strength of a hydrogen bond to a deuterium bond with just 2 lines of simple mathematics. It was wonderful. And at that time, I think it was true of most people who got into biology but coming from chemistry or physics, they had lousy ideas. They tried to superimpose some simple chemical or physical idea on biology. So I thought that somehow, maybe magic, if you could make creatures out of deuterium hydrogen instead of hydrogen 1, you could learn something. Don't ask me why that would make sense, but I think it was fairly typical of some people like me. To get into biology, you think of like making a mouse out of heavy water. Is that really crazy? Might learn something. Nevertheless, that's what I was thinking, that I would grow bacteria in D2O instead of H2O and maybe learn something, God knows what. So I'm thinking about heavy water. And if a great French biologist comes to Caltech and gives a lecture in Kirchhoff, this is Jacques Monod, and he and François Jacob, who later got the Nobel Prize for this, are asking how genes are controlled, what turns them on and off. I'm sure I've said this before.
Please.
Okay, well, I'm embarrassed. I don't remember what I said, but anyway, they discovered that if you add certain chemicals to a growing culture of Escherichia coli, namely lactose or certain analogs of the sugar lactose, like IPTG, isopropyl thiogalactoside, that the cells start making huge amounts of the protein beta-galactosidase. It makes sense because if the cell sees lactose, it needs to digest it, and beta-galactosidase is the enzyme that does that. So the cell is pretty smart. It doesn't bother making this enzyme unless it can be used, which means only if lactose or one of these other chemicals is like lactose is around. So the problem was, is the enzyme in the cell all the time, but just not enzymatically active, because the only thing that Jacob and Monod saw was the production of a lot of enzymatic activity. So there are 2 possibilities. Either the enzyme was there all along, but in an inactive state, and the inducer, IPTG, makes it active, maybe by bending it or adding a methyl group. The other possibility is the enzyme wasn't there at all until you add the lactose or the IPTG, and then the gene that makes it starts working, and you make a whole bunch of it. So one case it was already there, the second case it's synthesized de novo. how could you tell the difference? At this point, Monod, who's on the stage there at Kirchhoff, proposes an experiment to find out. And the experiment is to measure the change in the osmotic pressure inside the bacterial cell. God help us. I mean, maybe the bacterial cell wall is leaky, so you're not going to measure a meaningful osmotic pressure. See, if you make a lot of protein, which has a lot of groups that can bind water, then this water will want to come in. Let's say you have a piece of dialysis tubing, and you have a bunch of protein inside. Then water is going to want to come inside to bind to the protein. And so you'll have a higher osmotic pressure inside this piece of dialysis tubing. It's called the Donan effect. So that was the idea. And I'm sitting there in Kirchhoff thinking, this is the most god-awful idea I could imagine, because what do we know about the bacterial cell wall? Maybe it's got holes in it. Maybe, and how are you going to measure the pressure anyway? By the swelling of the cell. You can't conclude anything from this. I just thought it was awful. But I had deuterium on my mind because of the homework problem. And so I figured out, sitting there, that if you grow the bacteria, and since I'm thinking about heavy water day and night, you grow the bacteria in D2O, and so now everything, all the enzymes, including beta-galactosidase, instead of having hydrogens, will have deuteriums. And some of those hydrogens are not going to diffuse away. They're part of maybe CH bonds, not COOH. And so if you grow the bacteria in heavy water and then dump in the inducer, the thiogalactoside, and then quick like a flash, separate out the bacteria in a centrifuge and resuspend them in light water. If they have an enzyme that was made before you resuspended them in light water, that enzyme is made out of deuterium and not hydrogen. How would you tell if the enzyme was made out of the deuterium instead of hydrogen? You'd do it perhaps by layering the cell juice on top of a sucrose gradient and run the centrifuge and see how far down the enzymatic activity goes. And it will go down a little farther if it's got made of heavy hydrogen instead of light hydrogen. And you drip the stuff out of the tube by poking a hole in it and see when the enzymatic activity comes out. So that was my idea. And I go see Linus, tell him about this. I'm all excited. He says, good idea, finish your X-ray crystallography. I'm sure he didn't say it even that shortly. He must have found some very sweet, melodious ways. He was always so, he never said, do this. He always would tell you some story why you should do it and let you figure out that what he was trying to tell you was do this. So, but basically what he said, finish your crystallography. And then I went to Woods Hole, which is a wonderful place. You got to go there if you haven't. There are all kinds of biology courses in the summer. Don't go in the winter, it's too cold. And you can swim in the summer. And there I met my pal Frank Stahl. And if you haven't seen it, fish around on the web. There's a video of Frank and me, old guys by then, talking about Caltech, how it was the case that all the doors were open. Lots of members of the faculty knew about our social lives. It was a very close connection. I think it was partly, the student body was very small. I think there were 9 graduate students in all of biology. They were very small. There were more graduate students in chemistry, but I think there were only 9 in biology. It was a small place, and that meant that the faculty and the student body were very close, at least for those guys who had the nature of liking to be with young people. And that certainly was Pauling, and it was certainly Dick Feynman. So he used to come to our parties here, somewhere near here, just in front of that horrible-looking Beckman cake, wedding cake, was a street called San Pasquale. And Caltech obliterated the street. It's not there anymore. But we had a house, a big house that we rented from Caltech. and we lived there. And we had parties there, lots of parties. And Dick Feynman used to come and play his drums sitting on the carpet, and all the girls would sit around and admire Feynman and pay no attention to us. So in that respect, he was too friendly. But
is that how Feynman got involved in phage genetics and working with Charlie Steinberg?
I don't know how he, well, I already had got to know Dick very well for lots of reasons I won't bother you with. So when Sidney Brenner and Francois Jacob came to do the messenger RNA experiment, I must have told Dick that they were coming. So he asked if he could come every night when we'd have dinner to just talk about how the experiment was going. So every night we went to a place called Morton's Steakhouse, which is it still here? It was up on Colorado. A very dark, typical American place, a bar at one end and tables and dark. And so Dick was very interested in biology. And as far as I know, no, there was something before that. Dick wrote several books, and one of them was called See Here, Mr. Feynman. And in that book, he says he's walking to have lunch at the Athenaeum. By the way, do not call it the Ath. It's the Athenaeum. It's an important word. You're
Duly noted, Matt.
I mean, if an ancient Greek should appear here, you know, they had great big swords, and you said it's the Ath, they'd slice you up. Because there's a Greek. Anyway, so where was I?
Morton Steakhouse and Dick Feynman.
Oh, so after we did the messenger experiment, Dick continued wanting to work with messenger RNA. And we thought we'd isolate it. It's a rather boring thing to do, but we could determine its physical properties, molecular weight distribution, stuff like that. So to do that, you grow E. coli and then you grind it with alumina, because the crystals of alumina break up the E. coli cells in a mortar and pestle in the cold room, because otherwise your messenger RNA might get decomposed. So it's very cold and you're grinding away, and Dick did all this stuff. And then you put this stuff on top of a sucrose gradient, and you run the centrifuge, and you hope that the RNA, which is quite a dense kind of a molecule, will go down the bottom, and then you can get some nice pure RNA. Of course, there'd be other kinds of RNA there too, but why we were doing this, I can't really justify it. We were just doing it. And so then you'd get the RNA out by poking a hole in the bottom of the tube, and drip, drip, and now you got it. Well, we labeled the RNA with tritiated uracil, because we could identify it by its radioactivity. We put a certain amount of radioactivity on the top of the sucrose, run the centrifuge, poke a hole, let the stuff come out drop by drop, collect each drop, no radioactivity. Well, wait, we put it in. If you believe in conservation of mass, sort of fundamental idea, certainly Dick would have. So it had to be still inside the tube. For some reason, messenger RNA likes to stick to the inside surface of those, what were they made out of? Polyethylene tubes? I don't remember what they were made out of. They may have been nitrocellulose. So what do we do? Well, in those days, everybody knew that nothing will stick to Teflon. That's why you have Teflon-coated raincoats and all kinds of stuff. And the Caltech shop was so good, we thought, okay, they will make Teflon tubes. Now, they were kind of expensive, so you wouldn't want to poke a hole in the bottom. Besides, it's hard to poke a hole in a thick piece of Teflon. So we built a little thing that would collect the stuff from the top. A plunger with a hole in it connected to a little tube, and then the drops would come out from the end of that tube. And we had a little thing, you turn it, and it would drive the plunger down and down and down, and the drops would come out and drop, drop, drop. And we found that still the radioactivity would not come out, even sticks to Teflon. And then, you won't believe this, but we asked the shop to make stainless steel tubes. They did. I mean, that you had to have a furnace or something. They made beautiful stainless steel tubes. And again, we collected from the top. Nothing would come out. The radioactivity sticks to Teflon, it sticks to nitrocellulose, it sticks to stainless steel. So finally I decided it's got to be there, but let's, make sure that it hasn't become some quantum mechanical weirdo stuff. So we rinsed the tube with hydrochloric acid. And of course now all the radioactivity comes out, no problem. So it really was still there. And we finally solved the problem by having the idea, if we coat the walls of the initial tube, the kind of tube that you ordinarily would use, the nitrocellulose tube, with something else, maybe the RNA won't stick to the something else. And so we decided to try gelatin, because we had already some autoclave solution of gelatin. We rinsed the tubes with gelatin first, and then do the experiment. Works like a charm. The RNA does not stick to gelatin, but gelatin sticks to the walls of the tube very, very strongly and protects against losing the RNA. So why am I telling you this? God knows it's not very, it's not important that you know this. What is important? So sometime during your career, probably some of you will get mixed up with modification of the human genome. The people will want to make better athletes or flute players or cellists or God knows what by modifying the human genome. Now I want to warn you, be careful. Do you know what the interactome is? Have you ever heard the word? Raise your hand if you've heard of interactome. Good. So it turns out that most proteins in living cells do not act as monomers or even as homodimers or homopolymers. They act in very complicated ways. Most proteins make multimers with other proteins. And you can figure out which proteins bind to which proteins in the living cell by isolating carefully a given complex, and then with, hard to believe this, with a mass spectrometer, if it's a good enough mass spectrometer, you can get the precise, exactly, 0.00 out to any number of decimals, and from that you can get the atomic formula, because all the atoms don't have integral atomic weights. They're all a little bit off because of the packing fraction in the nucleus. And so just by having the exact molecular weight of a molecule, you can figure out what the molecule is. In other words, the empirical formula. And a whole lot of work like this has been done. And so the human interactome is now known to consist of at least 23,000 different protein complexes, other than just the proteins themselves. And there's probably some more to be discovered. Now, why is this important? Let's say you have a gene that will make it guys who better swimmers, better runners, or better flute players, something. And you believe that this one protein is going to make stronger people or something that you want. And you introduce it into the genome of somebody. But you don't know anything about the heteropolymers that might be formed. You don't know anything about what kind of an effect it will have in the interactome. In other words, you might lose something, or you might gain a function you really don't want. This is all very speculative, but it leads to the question of, if it's true that genetic engineering might cause you to lose something, or to have effects that you are surprised at, that you didn't expect, then you need to know what is it that's really important, what changes would be the most disastrous. So you could have different points of view on this question. Like you might lose eyesight, you might lose hearing, you might lose your little finger, who knows. But the worst thing to lose, some people might say, including me, is your humanity. I know this sounds kind of far off, but isn't that the most valuable thing we have? If we didn't have 5 fingers but still had our humanity? So how do you find out the genetic basis of, for example, compassion? Compassion, I think, is part of humanity. I hope. I think so. Here's how to do it. My daughter doesn't like this. So, at least for me, the Mozart Requiem is a very powerful composition. Now, I know there are other people who would rather hear something else. Hard rock, maybe. But you get 1,000 people, line them all up, and play the Mozart Requiem. And you've already got their genomes, everybody's genome has been done. And you put all these genomes into a big computer. Those are the people who liked the Mozart, who were moved by it, and those who didn't care. And you can now do what's called whole genome association analysis. The big computer will do that. And it will tell you which genes are characteristic of people who claim to be moved by the Mozart Requiem. Now, this sounds stupid, but if you can even begin, mostly, in most kinds of research, once you begin it, it's sort of stupid, but then as you keep on working, you learn more stuff and how to make it more relevant, more informative, and better. So my thought is that we should actually start this kind of thing now. It's real easy to do. You could do it right here. You could play the Mozart Requiem. Probably all your genomes have already been sequenced. Whose genome here has been sequenced? Oh, come on.
One. We have one.
No, really? Oh. Well, it will be. So it would be easy to do here. Then do it to the faculty. Get the faculty out, play the Mozart Requiem, ask the guys who are moved by it to tell you who they are. Ask CPG, chats, GPG, looking at all these genomes. What's the difference between the Mozart Requiem lovers and the guys who don't like the Mozart Requiem? And you'll get an answer, some kind of an answer. And if you begin doing all kinds of work like this, you could include, I guess, the Beatles. Pretty soon you begin to understand the genetic. Now, of course, there'll be an environmental basis too. If you never heard Mozart, you might react differently as the first time you hear it if you've been brought up with it, I know. And that doesn't change your genes, so far as we know. Listening, I don't think there's any genetic hazard in listening to Mozart. So I don't want to be accused of not believing the environment. But the genes must have some effect, I'm sure. And little by little, although you start with a stupid experiment like the one I've just suggested, if you keep on working on it, you begin to ask better questions and get more interesting answers. I think you should start it.
Well, Matt, we're coming up on the hour mark, so if anybody needs to leave, perhaps we can keep you for a little longer. Maybe there are some questions from the audience, but for now, if anybody has to leave, please feel free, and we'll continue the conversation as Matt's schedule allows. So yeah, let's take some questions. Do you want to?
Yeah, I have a question.
Very loud, my hearing's not too good.
Do you hear me now?
Yeah.
Yeah. Okay. I have a question about Sydney, actually, Sydney Brenner. And it comes back to my...
Sydney Brenner. About
Sydney Brenner.
About Sydney? Yes.
Yeah. Comes back to my undergraduate days in the other Cambridge, whereby if I was around those experts in protein synthesis, they would always make the rye...
repeat it to me.
You hear?
Speak a little louder.
They would always make the rye question, well, have you considered the magnesium iron concentration?
Oh, that's an interesting business.
Yeah?
But didn't I talk about this already?
No,
oh,
you didn't.
Okay.
So
we had already done a lot of work.
David has his
So my question was, going back to the messenger RNA experiment, what really happened to Sydney's inspiration to question the magnesium ion concentration?
It's more interesting than you might think. The experiment didn't work until the very last day because the ribosomes kept falling apart in the centrifuge, in the cesium chloride density gradient. And so nobody knew what to do. So the best idea was go to the beach and have a picnic lunch. And Hildegard Lamfrom, who was working with Jim Bonner here, made the lunch, and she had a car, and they all drove out, I think, to Santa Monica, and had a lunch on the beach. And Hildegard told Jim and told Sydney and Francois what her work was, and she was studying the in vitro synthesis of hemoglobin, and she was also doing an experiment involving the possibility of discovering messenger RNA, but by a different method. And she made the comment, by the way, the only way I know this, I wasn't there. I had left the day before to go propose marriage to my wife, so I wasn't there. But you can read about this in Francois Jacob's book called The Statue Within. And there he tells this story. They're sitting on the beach, and Hildegard is telling her experiment, and she says that she asked to add a lot of magnesium to get the in vitro synthesis of hemoglobin. And according to Francois's book, at this point, Sydney jumps up and yells, "It's the magnesium!" And they pile into the car and go back to Caltech, do the experiment another time with lots of magnesium, 0.04 molar, and it works. They learn it works the next morning because you have to run the centrifuge overnight. So the next morning, they drip out the tubes and they see it worked. And they leave that night, they're gone. So only a few people at Caltech knew that what had happened, and because they didn't hang around very long to tell people. But now, it's interesting why the magnesium. See, we thought 0.02 molar would be plenty. And that was because I had been working with ribosomes before Sydney and Francois came, studying them by sucrose gradient centrifugation, but also in cesium chloride. And what I had done so far up to that point was to show that if you treat ribosomes, actually an E. coli extract, with cesium chloride in a beaker, and then look at those ribosomes on a sucrose gradient to ask, have they been damaged by exposure to cesium chloride? The answer is they're just fine. What we didn't realize is that in the powerful centrifugal field of the centrifuge, proteins are much lighter than ribosomes. And if there's any protein that's in any kind of equilibrium with its ribosome going on and off, in the powerful centrifugal field, it will want to float up to the top. So we were shifting the equilibrium. The centrifugal force field was shifting the equilibrium of some of the ribosomal proteins so they would dissociate and go up to the top and give you not ribosomes, but ribosomes depleted of a few of the ribosomal proteins. We call them cores. But nobody at that time thought that it was the centrifugal force. If we had run the centrifuge slower, we might have got it all done within the first week instead of 3 weeks. But nobody had the thought that the problem is that the centrifugal field is tearing the ribosomes apart. But that's what was going on. Sort of neat.
Matt, what was Delbrück's reaction to the semi-conservative model of replication?
He was overjoyed.
How did that change his view on what Watson and Crick had proposed in '53?
Oh, no, our result was so crystal clear that...
But what did it mean? What did your result mean for Watson and Crick?
Our result meant that the proposal they had made for how the DNA molecule replicates is correct. Or at least more accurately, what we showed was that the DNA molecule is made up of 2 parts. What we didn't tell you what the parts were, but it was DNA we were working with. So, but in itself, if you just ask, what does this experiment tell you? It tells you these molecules are made of 2 parts, and when they replicate, the parts come apart and they become associated with newly synthesized parts. And that would leave the question, well, what are these parts? Your experiment doesn't tell you anything about what they are. And we actually considered this problem. It was an important problem. And especially we were worried about it because at Harvard, Paul Doty, who was a physical chemist there, using light scattering to measure molecular weights of DNA molecules. It's a very bad idea because for very large molecules, the light scattering is over a very, very small angle from zero. And so anything you get is likely to be wrong. They had concluded, though, that when you heat denatured DNA, there's no change in the molecular weight. That's wrong. But it's because the technique they used, light scattering, is very insensitive to molecular weight changes for very big molecules. We had shown that if you heat denature DNA, which is half hybrid, which is grown in heavy medium and then switched to light medium for one generation, that the two parts, the heavy and light, come apart in the centrifuge. So we knew that he had claimed that heat does not separate the DNA molecule into parts. And that's because light scattering is no good for that work. We had shown clearly that heat denaturation gives you two separate things because the heavy and the light chains far apart in the centrifuge. You could see that. But that still left the question, what are the parts? And that was done by two of my graduate students. One way was to do X-ray crystallography to try to measure the linear density of the DNA molecule. And that showed that it was just two-stranded and not four. And then John Cairns did some beautiful work looking at DNA molecules with the electron microscope. And he could see by measuring the length of the lambda DNA molecule and knowing its molecular weight, that it couldn't possibly be four strands, had to be two strands. So that little question at the end of our work, which was, what are these subunits that separate at the time of replication? They must be single polynucleotide chains, but you could argue and say maybe not. But what John Cairns did and what my graduate student did, it had to be the DNA molecule has only two strands, not four. So,
Question.
What was Linus Pauling's reaction to learning that that DNA doesn't have three strands but only has two?
What was Linus Pauling's reaction to learning that DNA has two strands?
joy. Everybody was very happy for two reasons. One, in itself, it's a beautiful result. Two, it was done at Caltech. You know, much of molecular biology came from here. I would start with the idea, one gene, one enzyme. That was Beetle. Now, we did some of it at Stanford, but they completed it here. So that was a very important step. One gene, one enzyme. And then you had Pauling's hemoglobin work, showing that sickle cell anemia is caused by a hemoglobin molecule that has an isoleucine where there should be a valine, or maybe it's an arginine, I forget. But anyway, there's a change in amino acid composition. That was a very important step in the whole development of molecular biology. And then you had our demonstration of how DNA replicates. Then you had the discovery of messenger RNA. There was a huge amount of work done at Caltech. The code, people were thinking hard about the code, but nobody here got it right. Max was thinking about coding, Bob Sinsheimer was, but of course it was Sidney Brenner and Francis Crick who got the code. And the thing about the code that's so interesting, the one thing that almost nobody thought about was it's read the same way we read English, from a starting point. So if you have a book, if let's say you have something, a book, in this rather simple story in the book, the story is cat, 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 cat. Well, because we know to start reading at the beginning, it's saying cat, 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 cat. But if you didn't know that, you might start with A, at, cat. Or you might start with the T, ta-ka, 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 ta-ka. And so far as I remember, nobody in all these ingenious ideas about the code said, maybe it's read like English. Well, that's a funny story.
What was the nature of your collaboration with Frank? Who did what?
Well, this goes back. So, okay. So after Jim and Francis got the double helix, Jim was obsessed with the idea that maybe RNA is a double helix, not just DNA. And the year after that year at Cambridge, England, he came to Caltech. Jim spent the whole year, 1954, here with Max Delbrück. And the reason he came, well, I think the reason he came wasn't just that he liked Max a lot, but because Caltech has these marvelous molecular models made in the shop. And I was here that year too. And so Jim was making molecular models of RNA, trying to find a way to make a double helical RNA. He never could do it. But there was a famous experiment done in England by a guy named John Gulland, G-U-L-A-N-D. And what he had done was simply to titrate DNA from neutrality to pH 1, very strong acid, and then ask how much base do you need to add to titrate it back down to pH 7. Now, if the molecule doesn't change at all, you need the stoichiometric amount of base equal to the stoichiometric amount of acid to go up and down like that. But if the molecule is changing in such a way as to present more titratable groups, then it might take more base to get back down from pH 1 to pH 7 than the stoichiometric amount equal to the amount of acid that it took to get up to pH 1. So Gulland found out that there was indeed just that kind of hysteresis. And that it must mean that there were titratable groups being liberated by exposure of the molecule to strong acid. And he also did the reverse experiment. So exposure to strong acid and exposure to strong base denatured the molecule. But not only that, but in the course of this denaturation, lots of titratable groups are liberated so they can titrate acid and base. Groups that had been hidden by being, and this was Gulland's idea, tied up in hydrogen bonds which don't titrate. So he proposed that there is a system of hydrogen bonds binding the parts of the DNA molecule. This was before Watson and Crick. And of course, when they got the structure, you could see these hydrogen bonds that bond the two chains. So Jim's thought was, he couldn't build a model, but maybe that was because he wasn't lucky. But maybe if there were two chains just like DNA, this titration experiment that had been done by Gulland on DNA would work out the same way with RNA. So he wanted somebody to do, because Jim didn't do experiments with his own hands, somebody to do this, and I was the guy. So I was living here in the Athenaeum, not the Athenaeum, and so was Jim, so we always had dinner together. And so he asked me if I would do this experiment, and I said, I'm going to go to Cold Spring Harbor. And he said, well, you could do it there, and I could pay your fees. And the way I could pay it is, we're going to call you a teaching assistant, but you're not going to do any teaching, you're going to do this experiment for me. So I'm a teaching assistant, except I'm doing no teaching. I'm working in the lab. Nevertheless, I met with, this is a long story, I met with the teachers, which was Jim and Sydney and Francis. They ran this course on the third floor of one of the buildings at Woods Hole. And at one point, Jim goes over to a window, and he points and he says, you see that guy down there? He thinks it's pretty smart. He used a special word that refers to the human anatomy, but that guy thinks he's pretty smart. Let's give him an experiment to see if he can do it. The Hershey-Chase experiment in one afternoon. That's an experiment that took L. Hershey and Martha Chase, I'm sure it took them a couple of days. So Jim is basically saying, let's give this poor guy an impossible assignment and make him realize he's not so damn smart. Well, I felt sorry for this guy, and I went downstairs, and there I find this guy, that's Frank Stahl. He's got a big jug of gin, and a bunch of tonic and limes, and he's selling gin and tonic to people who are walking by. I didn't realize that the main reason he's doing that is he didn't have much money and he was a big drinker, and some of the gin he drank. Anyway, so he's working on a problem involving bacteriophage. I'd never heard of bacteriophage, bacteriophage genetics. And he's trying to do a certain integral, a very simple integral of some exponential functions, and I knew how to do the integral, so I showed him how to do that. And then he's a much better mathematician than I am now, but in those days he didn't know much math. And then he told me about bacteriophages, and I told him my idea of how to determine how DNA replicates that I've already told you about, using the heavy labeled DNA and all that. We ended up using not deuterium, but as you know, nitrogen-15. It's better to use that. So we agreed, since he told me he was coming to Caltech, not the next year, but the year after that, as a postdoc, that we do this together. So a year later, he shows up at Caltech, and we share a house together. It was on San Pasquale, which you guys have destroyed. Wonderful big old house, lots of parties. And I say, let's start our experiment, our DNA experiment. And Frank says, have you finished your X-ray crystallography? Well, I had not. And he said, well, then we can't start the DNA experiment because it would be bad for your character. That's very much like Frank. He believes in character, which is, if you think about it, character is really important. Some people have got it and some people have less of it. Anyway, so then we finally began this experiment and it worked.
Did you need new instrumentation for the experiment?
We needed an ultracentrifuge. Now, there are two kinds of ultracentrifuge. One is called a preparative ultracentrifuge. It's used for making stuff. And the other is called an analytical ultracentrifuge. And it's called that because you can actually see what's inside of the cell that's rotating at thousands of revolutions per minute because of a complicated optical system and blah, blah, blah. So at first we started with a preparative centrifuge in the laboratory of Renato Delbrück, but we couldn't figure out, all kinds of bad stuff was happening. So we wanted to use a centrifuge where we could see what's happening while the centrifuge is running. And there was only two of those at Caltech then. One had been built here at Caltech, and everyone was terrified of it. So it was put in a special room with thick concrete walls because we were afraid it would blow up. Because, you know, go 50,000 RPM, it might blow up. And then there was a brand new centrifuge made by the Spinco company, which was owned by Arnold Beckman, who gave the money for that terrible wedding cake auditorium you got over there somewhere. And Jerry Vinograd, who was a senior postdoctoral fellow in the chemistry division here, had just bought one of these analytical centrifuges where you could see what's happening inside the rotating cell. And we asked him if we could use his centrifuge, and he taught me how to run the thing and gave us encouragement all along the way. So we included him. We wrote two papers, one about the development of this method of density gradient centrifugation, and he was a co-author of that because he'd shown us how to do it. And the second paper about DNA replication, that was just Frank and me. Anyway, if you look on the web, you'll find these two old guys, because it's a recent video that's been made, sitting on a sofa in Frank's house up in Eugene, Oregon. Lovely house. He's got two llamas in the backyard, reminiscing about Caltech. It's a really lovely video because you can get the feeling of what it was like, how nice it was from that video. So,
I think I'll ask one more question, Matt. How were you lured away to Harvard? Why didn't you stay with us?
well, I thought it was my duty as a patriotic American to see what the East Coast was. While I was growing up, the East Coast seemed like another world. It's way out there. From what I could tell, it was a different culture completely. And I thought that wherever I might end up, at least I should have the experience. I'd never been east of Denver, Colorado. Well, I'd been in Paris, but I'd never been in New York or Cambridge. So as far as I can remember, my motive was just to be on the East Coast, and especially in cosmopolitan part of the East Coast. I think more people now out here have, there's less of that, I think there's less of that, what would you call it?
Divide?
No. I forgot, there's a word for, well, I can't think of the word. We have a bunch of farmers out here. So that's the only reason.
And just as an addendum to that,
I thought, and by the way, there was more to it. Yes. Jonas Salk had invented the Salk vaccine. And people were really terrified of polio, that their children would get it. Nobody would go to the public swimming pools, were really afraid of polio. So when Jonas Salk invented the polio Salk vaccine, that was a big deal. And he decided he'd like to create an institute where good scientists would work on all kinds of biology problems. And he thought and he had a lot of money because people would give him money since he'd done this vaccine. He'd put it in Pittsburgh, which is where he was. And then Leo Szilard, who was a quantum mechanical physicist, he's the one who got Einstein to write the letter to President Roosevelt to see if we could make an atomic bomb, because otherwise the Nazis might get ahead of us. There's a whole story there. So Leo Szilard went to see Jonas Salk and told him no good scientist would ever go to Pittsburgh. You should put it in a beautiful place on the California coast, namely La Jolla. So Salk agreed with that, and they formed a little group of people, I was one of them, to create the Salk Institute. And there was a guy who owned a huge amount of real estate there near La Jolla, and he wanted something to be built there to attract people and business and income and all that, housing, shops, restaurants. And so he was all for having a new institute there that would attract people. And so he made a big chunk of land available, very low price. And we used to meet in a hotel in New York, the Carlyle Hotel. Leo Szilard, Jonas Salk, Francis sometimes, Milcone, all the initial people at the Salk Institute. And I'm supposed to go there too. And the architect who was, what's his name? So famous guy. I'll think of it. He designed my lab for the Salk Institute. But it would take a while. In the meantime, I thought I'd spend a little time at Harvard. But by the time they got the lab built, I decided not to leave Harvard. So it was more complicated than what I said at first. I did want to go to Harvard to experience that culture, but I was also marking time there before going to the Salk, but I never did go to the Salk because I got involved with a lot of politics down in Washington. And if I'd gone to the Salk, I'd have to be on an airplane all the time, and I didn't want to do that. And I'd made a lot of friends at Harvard and so on.
Matt, thank you so much for spending the time. Thank you.
I hope you'll never use the word "ath" again.